Thank you to Dr. Patel's insightful presentation on CMC. We are now going to switch gears and talk about the pharmacology and toxicology information that you will find in the Investigator's brochure, or the IB for short. As Leonard mentioned, I am Nikolett Biel. I am a nonclinical reviewer in the Division of Hematology, Oncology, Toxicology, supporting the Office of Oncology Drugs at the FDA. The learning objectives for this presentation are to be able to describe the types of nonclinical information provided in the IB and understand the relevance of this information to drug development. Nonclinical information includes pharmacology, safety pharmacology, pharmacokinetics and ADME. I will later touch on the meaning of ADME for clarification. Toxicology, which includes general toxicology, genetic toxicology, and other toxicology studies, including reproductive toxicology. Additionally, throughout the presentation, I will refer to guidance documents such as International Council for Harmonization, or ICH, and/or FDA guidances for industry that are available for drug developers to aid in the types and timing of nonclinical studies needed to support drug development. I will point to these references throughout the presentation and have also included a reference slide at the end of the presentation. In the nonclinical section of the IB, you will find information on the pharmacology of the drug. Let's take a closer look at what this information provides. The pharmacology section of the IB will provide data including proof of concept, or POC studies, demonstrating the mechanism of action of the drug, as well as its activity in vitro, for example, in cells, and/or in vivo, for example, in animals, such as anti-tumor activity in tumor-bearing mice for oncology drug products. The pharmacology studies will also demonstrate the primary pharmacology, or data on the intended target, and secondary pharmacology, or data demonstrating activity, or preferably lack of activity, on unintended or off targets. Furthermore, especially for biological drug products, the pharmacology studies can support the selection of species for toxicology studies. For example, some antibody drugs only bind human and monkey drug targets and may not bind the rodent target. In that case, the monkey will be considered the only pharmacologically relevant species to assess the safety of the drug. The amount of nonclinical data provided may vary depending on the type of drug product, whether a small molecule or a biologic. Biologic pharmaceuticals, such as immune-modulating antibodies, may have more pharmacology data, as animals may underpredict toxicities due to differences in sensitivity between humans and animals, or there may not be a pharmacologically relevant species for toxicology testing. So the safe starting clinical doses generally rely on many of the pharmacological data to determine what is called a minimally anticipated biological effect level, or MABEL. The amount of data may also vary depending on the indication. For example, pharmaceuticals intended for oncology or non-oncology indications. Additionally, the stage of drug development will also dictate the amount of data provided by the sponsor. Most pharmacology data will be submitted at the first-in-human stage, and additional data may be submitted to support different indications or combination regimens that may be evaluated during clinical drug development. Following the pharmacology section, you will find information on the safety pharmacology of the drug. Safety pharmacology studies are conducted to assess the potential effects of a drug on physiological functions on vital organ systems, including the potential for adverse effects on the heart, central nervous system, and respiratory system. Assessments on the cardiovascular system generally include in vitro assessments on the human ERG potassium channel and in vivo assessments mainly in non-rodents, for example, dogs or monkeys, including blood pressure and electrocardiogram measurements. Together, these data could identify a potential risk to normal heart functions. Central nervous system assessments include motor activity, behavioral changes, coordination, sensory or motor reflex responses, and body temperature. The respiratory system assessments include the measurement of respiratory rate. Central nervous system and respiratory system assessments are commonly conducted in rodents. Safety pharmacology studies can either be conducted as standalone studies, usually a single dose, or as part of the repeat dose toxicology studies. Both will use the intended clinical route of administration. Information on the conduct, timing, and exceptions for these studies can be found in the ICH S7 guidance for industry. Following the safety pharmacology information, you will find information on pharmacokinetics or PK and ADME, which means absorption, distribution, metabolism, and excretion. Pharmacokinetics is the drug's movement through the body as it passes through 4 different phases known as ADME. These studies describe how a drug moves throughout and is processed by the body. PK describes parameters such as maximal exposure, steady state exposure, the half-life of the drug, and clearance of the drug. These factors depend on ADME. Absorption is the process from administration, for example, taking a tablet, to systemic exposure or presence of the drug in blood plasma. The absorption can affect the speed and concentration of the drug in the systemic circulation. Distribution is the presence of drug in different tissues that may affect the efficacy or effectiveness and toxicity of the drug. Metabolism describes the different ways a drug may be broken down and identifies potentially human-specific metabolites, which would not be present in toxicology species. Excretion describes the mode of elimination of a drug, for example, through urine, feces, or bile. Toxicokinetics, or TK, is the study of ADME measured in a toxicology study with doses that may or may not be toxic to animals. TK informs on parameters such as dose proportionality and exposure. The exposure to some drugs is proportional to the increase in dose. For example, an increase by thirds in the dose would be an increase by thirds in the exposure. However, the exposure to other drugs may be less than or more than dose proportional. At times, exposure to drugs may reach a saturation point where increase in dose does not correlate to increase in exposure. TK can also inform on whether there may be differences in exposure between male and female animals, which can influence the determination of a safe clinical starting dose. TK also informs on the potential for accumulation of a drug or increased exposure to a drug upon repeat dosing. In some instances, for example, with biological pharmaceuticals, repeated exposure to the drug may lead to loss of systemic exposure due to the formation of anti-drug antibodies or ADAs that lead to faster clearance of the drug. Following the PK and ADME information, you will find information on the toxicology assessment of the drug. General toxicology studies prior to administering a drug to humans are conducted to determine whether the proposed clinical study is reasonably safe. Dr. Shabnam has touched on this during yesterday's seminar and how we use doses from the toxicology studies and animal to human scaling to determine a safe clinical start dose. The toxicology studies help determine a reasonably safe clinical start dose and guide dose escalation. They can identify potential dose-limiting toxicities that help inform the clinical safety monitoring and may assess potential toxicities that cannot be identified in the clinical trials. For example, potential acute histopathological changes in tissues and organs due to drug exposure. The toxicology studies use the same route of administration and schedule in the animals and intended in humans in the clinical study. Studies are generally conducted in two species, a rodent and non-rodent. The ICH M3R2 guidance describes the duration of clinical studies relative to clinical development of pharmaceuticals, and ICH S9 and S9 questions and answers is followed for oncology pharmaceuticals. For non-oncology drugs, the duration of the toxicology studies should cover the duration of the clinical trial. Studies of 2-week duration in two species support a clinical trial lasting up to 2 weeks. Toxicology studies supporting a clinical trial lasting between 2 weeks and 6 months should follow the same time frame as the intended clinical study. Studies in rodents of 6-month duration and in non-rodents of 9-month duration will support a clinical trial lasting longer than 6 months. For oncology pharmaceuticals to support a first-in-human trial, a study of 1-month duration in two species would support a trial, and additional studies of 3-month duration in two species are generally warranted to support a registrational trial intended to support a marketing application. For biological pharmaceuticals, there may only be one species for toxicology studies or none. I will discuss this in more detail on the next slide. Acceptable safety margins from toxicology studies to the clinical start dose depends on the indication of the drug, whether it's for an oncology or a non-oncology indication. The guidance estimating the maximum safe starting dose in initial clinical trials for therapeutics in adult healthy volunteers for non-oncology and ICH-S9 for oncology drugs can be referenced. Sponsors may use a variety of species to conduct the general toxicology studies. The commonly used species for rodent include mice and rats, and for non-rodent, the dog, monkey, and rabbit. Species selection may depend on the type of pharmaceutical, whether a small molecule or biologic. For small molecule drugs, rats and dogs are commonly used, and species selection may be based on comparable drug metabolism and types of metabolites present compared to humans. For biologics, species selection will depend on the target binding. Occasionally, only the monkey is a relevant pharmacological species because the antibody may not bind the rodent or other non-rodent drug targets. This is often due to minimal or lack of sequence homology of the drug target between human and rodent. In that case, toxicology studies are often conducted in the only pharmacologically relevant species. There are cases, although not common, where none of the animal species are pharmacologically relevant. In that case, a safe clinical start dose is determined based on the pharmacology studies. The next 4 slides include real examples where toxicities observed in animal repeat dose toxicology studies informed clinical monitoring. These examples are all from oncology, since I am a non-clinical reviewer in the oncology division. I do want to note that for oncology pharmaceuticals, we allow some toxicity in the animals to support a clinical dose in patients with advanced cancer compared to animal studies supporting a non-oncology indication. One example is a microtubule inhibitor drug developed for the treatment of advanced solid tumors. The drug led to irreversible optic nerve degeneration at the mid and high doses in the rat repeat dose toxicology study. Not only did this finding limit the dose from which the safe clinical start dose was calculated from, but also due to concerns the clinical monitoring was increased, including optic exams and imaging, and the information was added to the clinical protocol and patient-informed consent form. The second example is epigenetic targeting drugs that are being developed for the treatment of solid tumors and hematologic malignancies. The drugs led to malignancies or lymphoma in rat 3-month repeat dose toxicology studies. The secondary malignancy has also been observed in clinical trials. Based on concerns, the patient population being studied were considered, meaning the risk benefits were assessed and indications with indolent or less refractory tumors may have been removed from the trial. The information was also added to the IB and the patient-informed consent form. This information is also found in the warnings and precautions of the label of an FDA-approved drug in this pharmacological class. The third example is an antibody-drug conjugate, or ADC, with a topoisomerase I inhibitor. This ADC led to severe kidney toxicity, including increased urine protein, proteinaceous casts, and multifocal necrosis in the monkey repeat dose toxicology study. At the time of the IND review, due to the kidney toxicity in monkeys, the sponsor was asked to lower the clinical start dose and modify the dose escalation. Kidney toxicity was observed in patients during the dose escalation, which led to additional clinical safety monitoring, an update to the IB, and the patient-informed consent form to inform investigators and patients of the risk for kidney toxicity. In the fourth and final example, an immune agonist was being developed for the treatment of patients with urothelial carcinoma to be dosed locally in the bladder. This route of administration should localize the drug within the bladder and have minimal to no systemic exposure. In the monkey repeat dose toxicology study, a low level of systemic exposure was observed when monkeys were dosed locally in the bladder. The clinical team was informed that if systemic exposure is observed in humans, then there may be a risk for cytokine release syndrome, or CRS, with this drug. The clinical safety monitoring was updated to add up to 6 hours of monitoring of patients following the first dose in the event that systemic exposure may lead to CRS. Following the general toxicology studies, you will find information on genetic toxicology. Some drugs may be carcinogenic or have the ability to cause cancer by inducing tumors, increasing their incidence, or shortening the time it takes for tumors to occur. Results from carcinogenicity studies are generally not available until the time of product approval. To note, carcinogenicity studies are not warranted for pharmaceuticals administered infrequently or for a short duration of exposure such as anesthetics and radio-labeled imaging agents per ICH S1, and for advanced cancer indications, but may be needed for early cancer indications per ICH S9. In lieu of carcinogenicity studies, genetic toxicology assessments are used as a surrogate and help address concerns about the risk to humans during clinical trials. Additionally, results of genetic toxicology studies can inform on the duration of contraception. Genetic toxicology studies are generally conducted with small molecule drugs. A general battery of genetic toxicology studies includes 2 in vitro assays, one to test for gene mutation in bacteria, and another to detect chromosomal damage in mammalian cells. An in vivo assay commonly conducted in rodents to detect chromosomal damage to hematopoietic cells. Other genetic toxicology assays may be conducted. Further information can be found in the ICH S2 guidance. Relative to clinical development, the bacterial gene mutation assay is warranted to support a single-dose clinical trial. Additionally, the in vitro chromosomal damage assay is needed if proposing multiple clinical doses. The complete battery of studies, including the in vivo assay, should be submitted prior to a phase 2 trial. To note, for oncology pharmaceuticals, the genetic toxicology studies may be submitted at the time of the marketing application. However, results from genetic toxicology studies are needed to support clinical trials in healthy volunteers, which are typically conducted for food effect or drug-drug interaction assessments for oncology pharmaceuticals. Information on other toxicology studies in the IB include results from reproductive toxicology studies. Some drugs can cause teratogenicity or fetal malformations. One well-known example is thalidomide. This drug is used to treat leprosy and multiple myeloma, but was also prescribed to pregnant women for nausea and insomnia in the 1950s and 60s. The use of thalidomide in pregnant women in Europe, Australia, and Japan led to over 10,000 births with severe limb malformations. It was banned from use in pregnant women in 1961. Studies have shown that the sensitive period to thalidomide during pregnancy was approximately days 20 to 34 post-fertilization. The potency of the drug and the relative short time period between drug exposure and manifestation of side effects allowed the link between the two to be made. Reproductive toxicology studies are now a standard assessment during drug development to prevent side effects such as those seen with thalidomide. The timing of these studies depends on the patient population, whether patients are male or female, with or without childbearing potential. The timing also depends on the type of drug, whether it's a small molecule or a biologic, and the intended indication, whether it's non-oncology or oncology, and expected duration of treatment, whether it's short-term, usually the case with patients with advanced cancer, or long-term, such as years in non-oncology settings. Reproductive toxicity assessments are included in the general toxicology studies to inform on potential effects on fertility. Embryo-fetal pre- and postnatal periods generally follow the M3, S5, and S6 ICH guidelines and discuss the type and timing of these studies during drug development. ICH S9 is followed for oncology drugs, and generally only embryo-fetal toxicology studies may be warranted to support a marketing application. Some of these studies are not available at the time of the first-in-human clinical study. To prevent a potentially embryo-fetal or teratogenic drug to lead to adverse effects on the unborn fetus, clinical studies include testing for pregnancy prior to drug exposure and the use of effective contraceptive methods during treatment and for a period of time after the last dose of drug. In summary, non-clinical studies are an important part of drug development to inform on the mechanism of action of the drug, the safety of the proposed first-in-human clinical dose, the potential toxicities that may be observed during clinical studies, and inform on possible long-term adverse effects during the life cycle of the drug post-approval. Here I listed non-clinical resources and guidances that I have touched on during the presentation. Before wrapping up, I have 2 challenge questions for you. Challenge question 1. The safety pharmacology core battery does not include A, cardiovascular system, B, central nervous system, C, respiratory system, or D, the renal system. I will give you a few seconds to think about your answer. If you picked answer D, the renal system, you are correct. The core battery of safety pharmacology studies include assessments of the cardiovascular, respiratory, and central nervous systems. Challenge question 2. The complete battery of genotoxicity studies for non-oncology indications should be completed A, prior to the first-in-human IND stage, B, prior to initiating a phase 2 study, C, prior to submitting a registrational clinical trial intended to support drug approval, or D, at the time of the marketing application. I will again give you a few seconds to think about your answer. If you picked B, prior to initiating a phase 2 study, you are correct. If you picked D, at the time of the marketing application, you would have been correct if I singled out oncology pharmaceuticals, given the timelines for these studies vary depending on the intended patient population. That concludes my presentation. Thank you for your attention. I will be back to respond to your questions, but now I will turn it over to Dr. So to share her insights on clinical pharmacology.